Let's discuss what how they are structurally arranged and what are these nitrogenous bases, what are these deoxyribose sugar, and what are these phosphate groups. Okay, so let's discuss first about the nitrogenous bases. Okay, so nitrogenous bases, now there are two types of nitrogenous bases. We need to discuss what its structure is, so let's discuss it. First is purine, the other one is pyrimidine. Okay, so now what are these purine and pyrimidine? They are nitrogen containing carbon rings. Nitrogen containing carbon rings. Okay, and purine has two rings, pyrimidine has just the one ring. Okay, so now what are the names of these purines? Okay, purines include adenine and guanine. Okay, so there are just two purines. Okay, and there are three pyrimidines. So what are these? What are the names of these pyrimidines? Cytosine, thymine, not thiamine. Thiamine is a vitamin. Okay, so it is thymine. Most people say is thiamine sometimes, so it is wrong. And what is third one? Uracil. So pyrimidine is these three: cytosine, thiamine, and uracil, and purines are adenine and guanine. Okay, so what another thing, what are the things which are forming this purine and pyrimidine? Okay, they are formed by amino acid. Okay, so these two are formed by what? Amino acids. Okay, like these are the basic structure, like what is forming these nitrogenous bases from amino acid. These nitrogenous bases are formed. There are two types of nitrogenous bases, purine and pyrimidine, and they are formed by amino acid. And we all know how amino acids are formed. They are formed by the combination of elements, and amino acid is a type of molecule. Like what elements form these amino acid? Carbon and nitrogen. Basically, these are the elements which are forming the amino acid. And there are many types, more than 300 types of amino acids. So which amino acids are forming these nitrogenous bases? So there are only three essential amino acids which are helping in the formation of this nitrogenous bases. So let's discuss what are those nitrogenous bases. The first one is glycine, second one is aspartate, and the third one is glutamine. Okay, so these three are the amino acids: glycine, aspartate, and glutamine. These are the amino acids which are forming the nitrogenous bases, which nitrogenous bases purine and pyrimidine. Okay, so now we have got our nitrogenous bases. The first thing which was required for the formation of DNA, this was the first thing. So let's see which was the second thing here. The second thing is deoxyribose sugar. So let's discuss what is a deoxyribose sugar here. Second thing, deoxyribose sugar. So now what is a deoxyribose sugar? For that we must know that there are deoxyribose sugar is a pentose sugar. Okay, what does pentose means? Pentose means there are 5 carbons. Okay, any sugar which contains 5 carbons, it is called a pentose sugar. Okay, so now let's discuss how many types of pentose sugar are there. Pentose sugar, there are 2 types: ribose sugar, the other one is deoxyribose sugar. Okay, this is the second sugar. So what is the structure of this sugar? We already said pentose sugar means 5 carbons. Okay, so let's make those 5 carbons here. There's gonna be here carbon 1, carbon 2, carbon 3, and here is carbon 4, and here is carbon 5. Okay, and what will be in the middle here? Oxygen. Okay, and what will be here? OH, hydroxyl group. Okay, so there are 5 carbon, 1 2 3 4 5, and between them is oxygen, and this is the hydroxyl group. There are 2 hydroxyl group. So this is a ribose sugar. So what will be a deoxyribose sugar? We'll have oxygen here, first carbon, second carbon, third carbon, fourth carbon, and a fifth carbon. Okay, and there will be hydroxyl group. But what will be the difference? There in ribose sugar there are 2 hydroxyl group. So let's deoxyribose it. Let's remove one oxygen and hydrogen, just one hydrogen left. So what it will be? Deoxyribose sugar. Okay, so this is just a simple structure of deoxyribose sugar, and we know what it is made up of. It is made up of carbon, which is an element, 5 carbons, 5 atoms of carbon, and oxygen. The smallest thing in the world are the elements, uh, or the atoms of elements, and if we go into their detailed structure, then the smallest thing will be proton, neutron, and electrons, and much smaller than them are the quarks, which we are not really going to discuss. But you can say mainly the smallest thing in our body is an element bearing those atoms and their detail. So from these carbons and this hydrogen and oxygen, this deoxyribose is formed. Okay, so what was the third thing here? The third thing was phosphate group. Okay, so let's make our phosphate here. Phosphate group is nothing to discuss about, because it will have phosphorus and four oxygen, and phosphate will be formed. So this is just the phosphate. Now let's look at it here again. DNA is made up of nitrogen bases, deoxyribose sugar, and phosphate. Now these three should be combined. Okay, only then our DNA will be formed. Okay, so see how it is forming. This is our deoxyribose sugar. This deoxyribose sugar has to combine with the nitrogenous base. When it will combine with the nitrogenous base, then our DNA formation will start. So let's bring adenine here. Okay, this is deoxyribose, and adenine will come here. Adenine has arrived here. So what it has formed? It has formed adenosine. Okay, so adenosine is formed. Let's make the other ones as well. For example, this is another sugar, and here comes guanine. Okay, here comes guanine, and here comes cytosine, and with this sugar comes thymine. Okay, so now what we have here, when guanine combines with deoxyribose sugar, then what is formed? Guanosine. And same here, what will form? Cytidine. And what will form here? Thymidine. Okay, so this was the first combination that must occur. Nitrogenous base must combine with deoxyribose sugar. And what is formed here? These four parts, these four are a group of nucleoside. So now we have a nucleoside here. Okay, but what we needed, look at here again, DNA is a linear polymer of nucleotide, and these three should combine. So we have made nucleoside here but not nucleotide. So let's make this nucleoside a nucleotide. Okay, so for example, we have adenosine here. Let's take adenosine here. Okay, oxygen, carbon 1, carbon 2, carbon 3. Let's make the hydroxyl group here as well. This is carbon 4 and this is carbon 5. So, and let's bring our adenine here. So this is adenosine at the moment. This is just a nucleoside. So how to make it a nucleotide? Let's attach a phosphate group with carbon 5. So now this whole phosphate group is attached. So we have got all our three things here: nitrogenous base, deoxyribose sugar, and phosphate group. So where is our nitrogenous base, deoxyribose sugar, and phosphate group? So what it has become now? This is a adenosine, and these are the phosphates. So it is adenosine triphosphate ATP, and this is a nucleotide. These three has combined and it has become nucleotide. So what will happen to others? If others have this ribose sugar, here is guanine, and here is phosphate, then what will happen? GTP is formed. Okay, and again this is deoxyribose sugar, here is cytosine, and here is phosphate group. So what it has become? CTP. Again there is deoxyribose sugar, and again there is thiamine, the nitrogenous base, and there is phosphate group. So what it will become? TTP. So now all these ATP, GTP, CTP, TTP, all these three are what? Nucleotides. Okay, so this is a group of nucleotides. So we have got what? Our nucleotides here. Okay, let's go back. DNA is a linear polymer of nucleotide. What was included in nucleotide? This is actually a nucleotide structure. These three forms what? A nucleotide. Okay, so we have got our nucleotide clearly here. Okay, let's again recap it. DNA is made up of a linear polymer of nucleotide. Nucleotide includes